Hello and welcome to another Pype pod. We're going to continue with our anatomy physiology theme, and this time we're going to have a two-part series looking at the cardiovascular system. So jumping right in, a lot of you guys are aware of the content that's going to be passed on here. So what it's going to be is it's going to be a lot of core material, and then we're going to move on to adding a couple of new concepts and trying to link certain things that we know about the heart and the vessels and how they can be related to clinical considerations. Okay, so first of all, the cardiovascular system is an end-to-end network, okay, starting at the heart and ending at the heart. Okay, so you've got the heart and the blood vessel. No surprise, the heart is responsible for pumping the blood, and the blood vessels themselves are highways or mediums of transport all throughout the body. Part 1 will be focusing purely on the heart, and part 2 will be looking at the blood vessels and its correlation with the heart. The main function of the cardiovascular system is delivery. Okay, so whether it's delivering nutrients, building blocks like protein, amino acids, oxygen, it's the delivery system. On top of that, it takes things away from the body as well. So it removes carbon dioxide, it delivers waste products to excretory organs. Okay, so anything to do with movement around the body is primarily conducted by the cardiovascular system itself. And then secondary, obviously, the lymphatic system that we spoke about last time. So looking at the heart is located in the mediastinal of our chest. So that basically means mid-sternum, right in the middle of our chest. It's essentially where the great vessels and the heart lies. Now, the start of the heart itself starts at the second intercostal space. Okay, so pretty much just right under the second rib, behind the sternum. Now, the lower half of the heart is around the fifth intercostal space, and the apex of the heart is found to the left on the fifth intercostal space, and that's essentially the strongest part of the heart that you can feel, and it's also where you can listen to. The broad base of the heart sits on the diaphragm. Okay, so it's nestled on the diaphragm. Now, the heart itself is slightly to the left. Best way to think of the size of your heart, it's around the size of your particular fist. Okay, so that's how much room it occupies. If you were to open someone's chest up, it's majority covered by lung tissue itself. So you actually need to pull the lung tissue away in order to be able to see the heart itself. Now, the heart, like most structures in the body, are broken up in a series of layers. Now, these layers themselves have a unique function, and it's sort of biologically designed that way. So starting right at the top with the very most outer layer, we have what's known as the fibrous pericardium. Now, this works as a really strong protective layer that also binds it to the muscular wall. So the fibrous pericardium is the anchor for the heart itself, and it encapsulates the entire heart structure. Now, this, along with the pericardium that we'll talk about, is the last line of defense, stops the heart being able to grow too big or absorb too much blood, which we'll learn a little bit more about its capacity of holding blood later on. But this fibrous pericardium is responsible for its protective and the starting point of its fixed structure. The other things that help with its fixed structure and sort of holding all those loose semi-solid things in place is the pericardium itself. So the pericardium refers to the outer layer. And like most outer layers, it's up into two layers itself. So we have the parietal layer of the pericardium, and that sits just below the fibrous pericardium. Then we have a pericardial cavity and a visceral pericardial layer, which sits on top of the myocardium. Okay, now these pericardial layers are also known as the epicardium themselves of the heart. Now, the reason it has two layers is pretty much like why other structures in the body have two layers. So instead of having two fixed cellular structures together in an area that moves a lot, we have the ability and flexibility generated by surface tension between two layers. Now what I mean by that is the pericardial cavity is filled with fluid. So when the parietal and the visceral layer interact with one another, they're interacting as a result of surface tension. So as the muscles contract, the visceral layer of the pericardium allows and holds it in place, but it never actually touches the parietal layer itself. So there's always that surface tension and gap that allows for expansion and contraction and expansion and contraction without generating friction and cellular damage. That is the function of the pericardium. Now obviously things can go wrong. That's why we have pericarditis. And pericarditis is literally these two areas becoming inflamed either by an infection or sometimes it's unknown, but infection causes these layers to become bigger and then they start rubbing together. And if you listen to the heart, you will hear that rubbing sound. Okay, so that's the pericardium itself. Okay, the pericardium makes up the epicardium in layers. The next layer down is the myocardium. Now myo obviously meaning muscle, means this is the strong muscular structure of the heart itself. Now looking over on the picture, you can see that the left side of the heart, remember everything's backwards, is significantly larger than the right. Now I know that you guys were aware of that, I was just pointing it out, so then when we talk about circulation, it sort of makes more sense in how the myocardium works. Okay. And the last layer, or the inner layer, what's known as the endocardium. Now the endocardium is made of similar structures that is found within the vessels themselves. So it's a epithelial layer inside the heart that allows the blood to be stored in the heart without the transfer, if you will, of any fluids or any nutrients and things like that. So it's a protective inner layer of the heart. Okay. And then obviously the last area is the chambers itself. Okay, so the heart obviously has capacity, otherwise it wouldn't be able to do its function. So talking about the chambers of the heart, we know very well, hopefully, that there's 4 chambers found in the heart. So there's 2 atria, or 2 atriums, and there's 2 ventricles. The best way to think of the atria is that they are the receiving chambers of the heart, left and right, and they feed the ventricles, which are the discharging chambers of the heart. Okay, now each side of the heart is responsible for 2 different types of circulation. Now the problem is when you look at the structures of the heart, it's very simple to think about the heart as 2 independent areas, either 2 independent atriums, 2 independent ventricles, or a left and a right side independent of one another. Now that is just not the case. Whenever a contraction occurs, both the left and the right go together. Okay, so the atrium contract together, the ventricles contract together, and each aspect of the heart has a role to play in the overall cardiac output, which we'll discuss in detail as we go through how the functions of the heart and the structures of the heart assist in the overall outcome, which is cardiac output. Okay, so looking at the circulation, we mentioned the left and right side of the heart essentially feed different areas. So the right side of the heart is responsible for delivering blood to the pulmonary circuit. Okay, so this is the one that actually delivers blood to the lungs themselves. Okay, it's not that complicated. Delivers it to the lungs. The left side of the heart is responsible for what's known as the systemic circuit. So it's responsible for pumping to the rest of the body. Now, despite the fact that the left ventricle is larger than the right ventricle in muscle mass, the same amount of blood is pumped out to the lungs as to the systemic circulation. Okay, so the lungs get just as much blood to them as the rest of the body does. It's a fact that most people neglect to understand, is they think because of the size of the left that you're actually pumping more. The difference is not the mass, it's actually the force in which it comes out, which we will speak about. Now, Obviously, as these things go out, the whole purpose of going to the lungs is to get the blood itself oxygenated and decarbonated. Okay, that's the whole point. So the carbonated enriched blood will travel to the lungs and then get oxygenated before it comes back to travel to the rest of the body and then return. Now this is a never-ending cycle. So every time the heart beats, we get a fresh amount of blood coming back, and we have a fresh carbonated blood going to the lungs. And this gaseous exchange and swapping of ions happens every single second, basically, of every day. Okay, so that's a quick overview of the blood circulation, and we'll go through more of how the blood travels in certain structures in the heart. This is just a reminder to everybody, I'm sure. Now, what assists in this overall function of moving blood from one chamber to the next, not allowing certain chambers to mix with one another, optimizing the amount of fluid, if you will, or blood that is able to be pushed out, also known as stroke volume, is the heart valves. So the heart valves are uniquely designed, okay, and very specifically designed to allow or stop backflow. So they only allow for one direction of travel. Now the heart itself has 4 valves within it. It has 2 atrioventricular valves, or AV valves, between obviously the atria and the ventricles, and it has 2 semilunar valves between the ventricles and their respective arteries. Now on the right side of the heart, we have the tricuspid valve. On the left side of the heart, we have the mitral or bicuspid valve. Okay, both are absolutely correct. The difference is the way they're formed. Bicuspid has 2 flaps, tricuspid has 3. Okay, that's the difference. Now on the right side of the heart, we also have the pulmonary semilunar valves, and that's the valve in between the ventricle and the pulmonary arteries. And on the left side of the heart, we have the aortic semilunar valve, and that's obviously between the ventricle and the aorta. Now what essentially assists the AV valves holding in position is a structure known as chordae tendineae. Okay, so the left hand side is focusing on what's known as the preload side of the house. Okay, so essentially blood will come in to the heart via the superior and inferior vena cava. Now there's another one in there that fills it, but we'll talk about that one later. So it comes in and it starts filling up the atrium. Now the AV valve is not going to open automatically as soon as blood gets in there. There has to be a certain amount of pressure before they will open in order to fill the ventricles. Okay, so don't think you need 10 mils or something like that and all of a sudden they open up to fill the ventricles. It's just not the case. Okay, so a certain amount of pressure is needed in the right side, filled by the inferior and superior vena cava, and on the left side of the heart, the left atrium is filled by the pulmonary veins. Okay, just the pulmonary veins come in, the 2 come in and they fill the side. Now as the pressure builds up, the AV valves will essentially open down, okay, and this allows for ventricular filling. Okay, once ventricular filling occurs and the ventricles start to feel pressure within themselves, they'll start to pre-contract. Now we'll talk more about the cycle later on, but once the pressure in the ventricles build up, they close the AV valves themselves, and the chordae tendineae stop them from inverting themselves into the atrium. Okay, so that's how the AV function works. AVs open down from the atrium into the ventricles, and the chordae tendineae stop them from inverting the other way. Once the ventricles contract, so once there's enough pressure in it, the semilunar valves open up, allowing the blood and the pressure to pump through the arteries, pulmonary artery, aorta itself, pumps through those and the semilunar valves open up. Once the main pressure has dissipated or has equaled something known as afterload, then the weight and the pressure of the blood itself collects in the cups of the semilunar valves, and the semilunar valves will actually close, filled with blood in small cups, and stay closed because of their own internal surface tension. So they will not be able to invert the other way under normal circumstances because of the pressure associated with the blood and the structure in which the semilunar valves have. And that is how the valves work in order to assist in the overall function of the heart. Now, this coronary circulation that I was speaking about before is the concept of feeding the heart itself. So whilst the heart contains a huge amount of blood, and half of the heart has a very enriched oxygenated blood, the heart itself still needs its own circulation in order to survive. Now, what is more incredible, if you ask me, is that the arteries and the vessels associated with the heart, every vessel in the body, also needs its own circulatory supply. So it doesn't get its nutrients from itself. Now, the capillaries do, okay, they're a little bit unique, but every other vessel needs its own capillary network in order to nourish itself. Okay, but that's a side note because I think it's cool. Now, when we're talking about the coronary circulation, we obviously have things called coronary arteries and coronary veins. As the left ventricle pumps and it moves into the aortic arch, part of that blood is diverted into the heart and it feeds the coronary arteries. And then it gets travelled back towards the right atrium by the cardiac veins, and then it empties into the coronary sinus. Okay, so now we have the inferior vena cava, superior vena cava, and coronary sinus that actually fills the right atrium itself. So that blood doesn't go anywhere else. It feeds the heart and then get pulled back into the right atrium. Okay, that's how it works. When you think about it, the heart is a muscle and it is working harder than any other muscle in your body. So it needs a constant and more rapid supply of oxygen in order to feed itself, in order to generate the energy needed to contract. Okay, so that's why we have this special network, closed network of circulation around the heart. Now, speaking of how amazing the heart is, we'll look at the cardiac muscles themselves. Now, cardiac muscles are the most unique muscle structures in the body. They look a little bit like skeletal muscles, okay, but they have completely different characteristics to any other muscle in the body. So they're striated, they're quite short, they're a little bit chunky, but the biggest thing is they're interconnected. Okay, so when you get skeletal muscles, they're connected to one another. Each myocyte is connected to one another and it follows along a filament only. So the impulse follows along one filament and that is it. What happens in the cardiac muscles is we have things called intercalated discs. Okay, and you can see them on this lovely stain that we have here. Okay, so the intercalated disc is the junction point and the anchors of the heart itself. Now, we talked a little bit about the heart having the ability to stretch. Now, I'll just mention it here. We will mention it later on, but there's a very, very clever man that wrote a lot of good books called Frank Starling. Okay, and he essentially made a law up about the heart. He said, with the greater stretching of the cardiac muscle, the greater the contraction will be. Okay, so that's Starling's law of the heart. Now, the cool thing about that is the cardiac muscles themselves have the ability to stretch. So at rest, their actin and myosin are not quite lined up beautifully. But if you were to fill the heart, allowing the muscles to stretch pre-contraction, you'll actually find the actin and myosins will line up better, creating a better contraction when it happens. Now, what prevents the cardiac muscle falling apart from all this internal pressure and contraction is desmosomes. Now, desmosomes are connection points between the cardiac striations themselves, and it holds it in place. The bonds are very, very strong and they hold them in place. The other cool thing about the intercalated discs is we also have gap junctions. Now, normally when we're dealing with action potential, ionization, depolarization or contraction, whichever one you want to say, they're all pretty much the same, is you have it in a linear fashion. It will go from one end, will activate more sodium channels across the chain all the way to the other end, and that is it. So muscles will contract along the striation. Cardiac muscles are excitable. They have the ability to pass ionized charged or trigger increased amperage to start action potential along its entire axis, okay, which means almost like a ripple effect. So it has the ability to pass charge, ionized charge, to different striated structures. Okay, and that's why we can have that perfect contraction as it goes around, because it's not lineal, it essentially wraps around the heart itself. Now, when you just look at the cardiac muscle of the heart, you'll actually notice that it's intertwined around the heart. So almost like a figure of 8. It starts at the atrium and wraps around in an arc, and then the muscles travel down the ventricle and then intertwine like the infinity symbol at the base of the apex. Now that structure allows the heart to contract together and then also twist or almost squeeze like a sponge in order to get most of the blood out of the ventricles. It almost has a twisting contraction. Now the reason that occurs is because of these intercalated discs and gap junction, and obviously the ionized or the electrophysiology of the heart itself. So the conduction system of the heart is amazing. Okay, it's an intrinsic conductive system, or it's also known as a nodal system itself. So we have specialized structures within the heart that allows the passing of ionized charge along optimal pathways. Okay, now each one of these areas in the heart has their own unique automaticity. So autorhythmic cells is what they mean. So essentially they can be stimulated by external source, or they can just react themselves based on their own leakiness of sodium. Okay, so the sinoatrial node is obviously the pacemaker of the heart. That's where you get a sinus rhythm. That's where the impulses start, ideally. From there it passes to the atrioventricular node, down bundles of hysts, and then around the Purkinje fibers. Now that pathway that it travels allows for optimal contraction. Now this is the least I've ever spoken about the conductive system in my life. I'm not going to talk about any more, because if you guys want to know absolutely everything about conduction and the heart system that you'll need to know, then it's back to episode for conduction and ECGs. So I'm not going to labour this presentation with too much information on that, just only to let you know that that information is there. Okay, that system is there and it all works together. So we're going to move on to something else, the cardiac cycle. Now a cardiac cycle means after the atrium have contracted, they will relax and then the ventricles will contract. And then there'll be a little bit of period and then the atrium contract and it continues that way. Now the contraction aspect of the heart is known as systole, and the relaxation aspect of the heart is known as diastole. Yes, I know you've heard of these terms before. They're associated with blood pressure and with good reason too. So looking at the cardiac cycle itself, okay, it's broken up into 3 areas. So we have the first area. Now number 1 starts at mid to late diastole. Now realistically, you know this is a continued loop, so you could have started everywhere, but for ease of learning, it's best to start at mid to late diastole. This is the time period that the ventricles are filling. Okay, so we've had the atriums have filled enough in order to open the AV valves, so now we start getting ventricular filling. From there, the pressure is going to fill up in the ventricles, and they're going to be 70 to 80% full. Now it depends what book you read. Okay, most say 70, some say 80, but they're going to be 70 to 80% capacity. From there you get atrial contraction, and this is the part that it injects the extra 20 to 30% of blood volume into the heart itself. Now this engorges the ventricles, which leads us into the second phase, which is ventricular systole. So initially, before we get the ventricular systole, we have an isovolumetric contraction phase. Now this is that Stirling effect that I was talking about before, where the heart is now stretched and it's building itself up to capacity. So it has massive resistance against it and it is ready for optimal contraction. Now the reason that we don't have the semilunar valves opening up is because of this concept known as afterload. There is pressure in the arteries themselves, and the ventricular pressure has to be greater than that in order to open those semilunar valves. So isovolumetric contraction phase doesn't usually get high enough. So when you get the contraction side of the house of the ventricles, it opens up the semilunar valves, and then all of a sudden they empty themselves as much as they can. The third phase, early diastole, you're going to have a little bit of blood left in the ventricles, okay? Unless, of course, it's been triggered by something that triggers its contractility, which we'll talk about soon. Then you can have a little bit less, okay? But the early diastole is where the atrium start filling themselves. Okay, so there's a thing in the ventricles right now. The atriums are starting to fill up, and then we go back to stage 1. And this cycle continues. And all of it has to do with cardiac output and how to optimize cardiac output itself. Now, during this cycle, you obviously, if you listen to the heart, you will hear heart sounds. Okay, now we've all heard of the lub-dub side of the house. Okay, now how much you like that or not, it's irrelevant. It's what people say. Okay, so what the lub-dub actually is the closing of certain valves. Okay, that's it. Okay, so the lub side of the house is essentially the closing of the bicuspid and tricuspid valve. That's it. As they close, they make a lub sound. And then as the semilunar valves close, the aortic and pulmonary semilunar valves close, it makes the dub sound. Okay, now the mitral, tricuspid, AV valves. Closing is known as S1. That's sound 1, if you will. Okay, sound 2 is the semilunar valves closing. Okay, and they're normal. You'll hear them all the time. Now, clinically, you can get additional sounds that can mean some form of pathological state. Okay, so S4 is a sound where you get atrial contraction and the ventricles aren't ready to deal with that, with the additional load, basically from there. So the atrium either contract early or the ventricles aren't as full as they should be, so we get this vibration sound against the walls themselves. Okay, and that happens before S1 sound. So obviously before the AV valves have closed, you can hear this additional sound, which essentially means non-compliance between the atrium and the ventricles. Okay, S3 is the other end of the scale. Okay, this is what happens when we've pushed the ventricles out, then all of a sudden we're starting to get refill too early. So you're getting refill straight after the actual closing of the semilunar valves. Now the reason that's important is because sometimes it means that the AV valves are loose, opened up, so you're getting pouring into the ventricles too early, so you hear it. Or it could mean that the afterload is too great and the semilunar valves are broken, so you get that backfill from the arteries. Okay? Either way, it's a pathological thing that needs to be looked into. So that's the different sounds that you can hear, S1, 2, 3, and 4, and where they fit in the whole cycle. Now, as I mentioned, the outcome of all this that you've learned is all about cardiac output. Now, cardiac output literally means how much blood is pumped around the body in a minute, and it's calculated by heart rate versus stroke volume. Stroke volume means how much blood is pumped out of each ventricular contraction. Okay, and on average it's around 70 mils, just to let you know. Now, there's obviously things that can affect cardiac output. Now, the easiest thing to change is the heart rate itself. Okay, so you can change the heart rate pretty easy, and it happens all the time based on need. But the stroke volume can also change, and it's got to do with 3 factors and the compliance of this Starling's law that I've been talking about the whole time. Okay, so that's the two factors that can influence cardiac output. The easiest change is the heart rate, but we can change the stroke volume in order to have a better outcome. So let's break it down just nice and quickly. So when we look at increasing the heart rate, what that means is there's going to be more beats per minute. So regardless of what the stroke volume is, if you increase the number that you're timesing it by, you're obviously going to end up with a larger number. Okay, that's obvious. Now, things that control the heart rate is autonomic intervention or hormonal intervention. Okay, and it comes mostly as a feedback system. So you need a higher heart rate and you get a higher heart rate. The autonomic intervention is all about your sympathetic drive. So your sympathetic drive is a central nervous system, sympathetic fibers. They will be found in the heart itself. So they will be connected in the SA, in the AV, in the myocardium itself, and also in the arteries as well. So that's where you'll find sympathetic nerve fibers. Parasympathetic nerve fibers are essentially found in the SA node. That's it. Okay, parasympathetic fibers are associated with slowing the heart down and overruling the sympathetic fibers. Sympathetic fibers increase heart rate and a little bit of afterload, things like that, because of vasoconstriction. The other thing that aids in heart rate is linked with the autonomic intervention, and that's hormones. So adrenaline, noradrenaline causes your heart rate to increase, and it causes vasoconstriction. Now, thyroxine as well is another hormone that increases your heart rate and causes vasoconstriction also. So that's the factors influencing the heart rate. Now, when we look at stroke volume is broken down into three different areas. So we have preload, contractility, and afterload. Those three things influence the overall stroke volume. Now, preload is all about how much blood is getting into those atrium. So it's got to do with how much blood is coming back within the venous return and how much time is left for the atrium to fill. So if someone has a high heart rate without a real need, then you're going to have less filling time and a reduction in the preload, meaning you're going to have a lower end diastole volume, and it's going to influence the end systole volume because it wouldn't have pushed in that extra 20 to 30%. Now, contractility, we talked about. Contractility is associated with the same things as the heart rate. So sympathetic drive increases contractility, and then we also have hormonal changes. So adrenaline increasing venous return because of vasoconstriction, which taps into that Starling's effect. More blood in there, you're going to get a greater contraction. That's how that works. Now that's just got to do with your end systolic volume. Now afterload is a little bit tricky. Okay, afterload means the pressure associated with the arteries closest to the heart. So this is influenced by vasodilation and constriction. Obviously, if you reduce the pressure in the arteries, like the aorta and the pulmonary arc, then you're going to allow for more blood to come out next time. Now, the way that works is just imagine if you have somebody with a normal blood pressure. Okay, so 120/80. That essentially means that 120 about is going to be right at the heart. Okay, so you've got 120 millimeters of mercury at the arteries. That means that the ventricle pressure has to be greater than 120 in order to push out the optimal amount of fluid. Now the problem is as soon as the pressure gradient changes, as soon as the pressure from the ventricle drops below 120, the valves are going to close. Okay, because there's greater pressure on the other side of the vessels. Okay, and that's how it works. Now if you're hypertensive, so have a really high blood pressure, then you're going to have a higher afterload. So the higher the afterload, the lower the stroke volume you can have. Okay, and that's the factors that affect stroke volume. Stroke volume is made up to influence cardiac output. Now, when we move on to the next lesson, we'll look at the vessels, and we'll put all these together with how cardiac output can assist in body functions from there on. Okay, well, that's part 1 done. Like I said, we're going to move on to vessels next presentation, and hopefully bring it all back in together. Now, I hope you got something out of it. Now, I do apologise if a lot of it was suck eggs. We do spend a lot of time looking at the heart itself, but hopefully it enlightened at least something. Okay, so until next time, take care.